Hey guys, how are we doing? Hopefully your weekend was good. Because welcome back. We're going to dive right into talking about the cell. Okay, so I'm going to share my screen, and we're going to go over this PowerPoint. Maybe if I can think about what I'm doing. All right. Okay. So let's go ahead and look at um, the pre-lab assignment, although let me move some stuff out of the way that you can't see first so that I can see. There we go. Okay. All right. Categories of cells. So basically in the pre-lecture assignment and in the reading that you would have done, and as always, if you haven't done it, please stop and go back. It makes it a whole lot easier for you and me. You would have gone over the different types of cells. You would have talked about, let's try that again. All my electronic knickknacks to make this happen. You would have talked about prokaryotic cells. That was terrible. What even is happening with me? Just do that. Prokaryotic cells and eukaryotic cells. Okay. So prokaryotic cells overall, they're smaller. They lack organelles. And think about things like bacteria. Really, bacteria are the types of prokaryotic organelles, or cells, forgive me, that exist on Earth. Okay, so prokaryotic cells lack a nucleus, and this is the big distinguishing characteristic, whereas eukaryotic cells have a nucleus. Okay, so we are made out of tons of eukaryotic cells. Okay, so again, an example of a prokaryotic cell with a nucleus right there, which stores our genetic information. Okay, in a prokaryotic cell, the genetic information is just stored loose in the cell, if you will. However, there are some things that prokaryotic and eukaryotic cells all have in common, and they're just kind of characteristics of cells in general. The first of those is the plasma membrane, which we talked about a little bit in a previous lecture. So the plasma membrane is this outer barrier, goodness. Remember the phospholipid bilayer? Had the phosphate heads and lipid tails, the way it was formed, okay? So that's one of the things that all cells have in common. The phospholipid bilayer is selectively permeable, which I'm going to write up here above it. Selectively permeable, meaning only some things get through. Okay, some things get in, others don't. Another thing that both eukaryotic and prokaryotic cells have is the cytoplasm. Consistency of jello. So organelles can be suspended in it, kind of like if you make jello salad with fruit in it, which by the way, no thank you. I'm just gonna say that right now. Anyway, but the fruit in a jello salad would be like organelles that are suspended in it. And then finally we have, I'm just like now looking at how this is worded. This should not have been worded like this because now this throws it off. What am I gonna do here? I'm gonna draw a line. Because eukaryotic cells and prokaryotic cells both have plasma membranes that are selectively permeable, and they both have cytoplasm. But they also have genetic information, at least one chromosome, and they have ribosomes. So these are the 4 things that they have in common. So this was kind of accidentally, I think, worded incorrectly, because eukaryotic cells are the ones that have what we associate with organelles. The mitochondria, again, the nucleus, endoplasmic reticulum, the Golgi apparatus, all of these different things. Prokaryotic cells aren't going to have that. They're only going to have ribosomes. Okay, so organelles are internal compartments that have specific functions. And it's those functions we'll kind of go over as we go through this particular lecture. Specific, actually, I think the text might have said specialized, but same difference. Spell it. Okay. All right. Let's go ahead to the next one. I gotta do all this fancy stuff to be able to advance a page. There we go. All right, so let's talk first when we're talking about semi-permeable membranes, right? These membranes that let some things out but not everything, and some things in but not everything. We have to talk about some passive transportation or passive transport. Passive transport means that there's no energy required to make it happen. As you might imagine, the cell wants to use as much passive transport as possible. It doesn't want to use up the energy that it's creating. That was a big crash. Okay. So one form of passive transport is something called diffusion. In diffusion, molecules move from a high concentration to a low concentration, okay? And that path that they follow is called a gradient, okay? So they go down a concentration gradient from where it's super crowded to where it's like less and less and less crowded, okay? All right, before I continue, here's how I think about diffusion. Molecules, atoms are a lot like people in the sense that they don't really like to be crowded. If we were meeting in a classroom, okay, picture one of the rooms at the university, and there's a good number of us. I don't know how many are in this, and what am I at? 60, 70? So let's say that we are all in this classroom together, and I tell you, all of you have to get up and go stand in a corner, at the same corner, and you have to pack in as tightly as you can, okay? So picture that for a minute. Got to go squish into a corner with 60 to 70 other students as tightly as you can. You're probably not going to be too happy about that. Neither are atoms and molecules. They want their personal space, much like you do. So as soon as I were to tell you, okay, you can walk, you don't have to stand there anymore, you guys would spread out, probably to take up as much of the classroom as possible and sit as far away from each other as you can normally. Even without COVID, people do that. Anyway, again, molecules and atoms do the same thing. They want to go from where it's like super packed and crowded to where it's way less crowded. That's a concentration gradient. Really crowded to less crowded, that path is a concentration gradient. And diffusion itself is going from high concentration to low concentration. Again, kind of a concept you already get because people are like this too. Another type of passive transport is a process called osmosis. And osmosis is diffusion, right? In the sense that we've got high concentration to low. But what it's talking about specifically is the movement of water molecules. And that is a huge, what do I want to say? I can't think of the word. This is what makes it different from just diffusion. So like the words diffusion and osmosis cannot be used interchangeably. Because if you use the word osmosis, you're only talking about the movement of water and nothing else. Okay? Movement of water across a semi-permeable membrane. Oh my goodness. I think my kids are unloading the dishwasher, and all I can do is hope that when I get out of here, I'm still going to have things that aren't broken. Okay. So when we're talking about osmosis, we have to talk about a new term. That I'm going to take a second to try to explain even before I fill stuff out. We're going to talk about solutes and solutions. So I think I'm going to switch here to my little whiteboard. Maybe, there we go. Move stuff around as usual, give me a second. So we have this term solute, and a solute is something dissolved in a liquid, usually water. I mean, the liquid is usually water. Okay? And then we have the word solvent. Solvent is something doing the dissolving. And so I want to switch that to not something. I'm going to switch it to the liquid. And again, that liquid is usually water. Let's abbreviate it this time. Usually water doing the dissolving. Okay. Then when you have a solution is this ratio, or the ratio of solute to solvent. It's, I'm just going to simplify it here, a mix of solute and solvent. So let's do an example real quick. Because you're really familiar with these terms, you just don't know it because it's not what you walk around saying to yourself. But if you've ever had Kool-Aid before, right? Kool-Aid, isn't that how you spell it? Something like that. If you've ever had Kool-Aid before, you know that Kool-Aid is a mix of powder, Kool-Aid powder, and water. Okay, so let's label it. The powder, you tell me what it's going to be first. Answer it. Is it solute, solvent, or solution? So the powder is going to be the solute, right? Because this is what we're going to dissolve in the water, which means that the water is the solvent. And the Kool-Aid then is the solution. Okay? So that's how that works. How about Tang? Should we do Tang? I always forget how much I like Tang until I have it for some random reason. Then I'm like, oh yeah, I liked it. Hey, powder plus water, same difference. Same thing, solute, solvent, solution. Okay. So I want you to keep thinking about, we're gonna go with Tang. I want you to keep thinking about Tang for a minute. This would work with like lemonade too, if you're thinking about powdered lemonade. We're just gonna go with Tang. You know that you can make Tang either really strong, right, where it has just like a ton of flavor, and maybe even like some Tang powder that settles to the bottom because there's so much of it. Or you can make it kind of just in the middle, where it's pretty balanced. Or it can be nastiness, where it's weak. In other words, you barely get a little bit of the Tang flavor, but mostly it's just water. Everybody knows this, right? Every kid, when they start making Tang or Kool-Aid or lemonade, knows that you can change the amount of powder you put in to the water to kind of decide if you want it to be really strong or really weak or just perfectly in the middle. Okay, well, these solutions of Tang have names, vocabulary words, if you will. Something that's really strong has a lot of solute, or lots of Tang powder. That's our solute. Such a solution is called hypertonic. This term hyper means above or more, and it is talking about the solute concentration. Something that's hypertonic has more solute, okay? Now, if it's really weak, then it has less solute. I'm just going to put little solute. And that word is hypotonic, meaning below or less than hypotonic. Okay? And again, the prefix hypo is referring to the amount of solute. Hyper, above, so it has more solute. Hypo has less solute. In the middle is something called isotonic. An isotonic is a balance of solute and solvent. Okay? Now, when I say it has lots of solute or little solute, that means it's in compared to the solvent itself. So if I have a hypertonic Tang solution, I've got more powder in there than I have water, which is why you can see the little Tang powder particles drift to the bottom. There's not enough water to dissolve them. When it comes to hypotonic, I have way more water than I have Tang powder, which means I could add more Tang powder to it because there's enough water molecules to dissolve it. Isotonic, it's, you know, you can think about it kind of being as like on a 1-to-1 ratio. There's the same amount of Tang powder as there are water molecules, okay? So this is tonicity, and it has to do again with solute, solvent, and solutions. Okay, having said that, let me go back over here to my PowerPoint. We used Tang. The PowerPoint is going to talk about red blood cells. Okay, I've already given you diffusion and osmosis. So I'm not going to fill those out again, but we'll come down here to our part on tonicity. Sorry, give me always 1 second when I have to switch between screens. Okay. So when it comes to water moving, water follows the solute. Okay, let me put that here. Water follows the solute. All right, that means that water is going to go from a low solute concentration to a high solute concentration. This is the opposite when you explain it this way of what you think with diffusion. That's because, let me see, these don't really have the best images to go with this, but essentially if you have something that has a little bit of solute over here, and then you've got a ton of solute over here, the water is actually going to go from where there's less solute over here to where there's more solute. Why does it do that? It's in an effort to bring balance, okay? In living systems, it's always about balance. Everything likes to be within certain parameters, certain ranges to function optimally. Fortunately, our bodies have ways of keeping ourselves there. And one of the ways is this process of osmosis and of water going from where it's not as crowded with solute to where it's way crowded with solute. And all of that, again, to try to balance it out, okay? So there's as much solute in one part as there is on the other, okay? So this is the same vocabulary I gave you on the slide. Hypotonic has low solute. This is an example here of a hypotonic solution. With red blood cells, what happens is inside the red blood cells themselves, there's a lot more solute. Sorry, a lot more solute than there is out here, okay? That means that water wants to balance this out. So it wants to go from outside of the red blood cell inside the red blood cell. The problem is red blood cells can only take on so much water before they burst, similar to a balloon, a water balloon that you fill with too much water, it pops. That's what happens in a hypotonic solution to a red blood cell. It eventually takes on so much water because it's trying to balance out the mix of solute to solvent that it will rupture and burst. Rupture and burst, because there's only so much water it can hold. Okay, and a hypertonic solution with high solute, this is it over here, the opposite happens. Here, there's a ton of solute out here in this surrounding fluid, okay, in the fluid that's surrounding the red blood cells, tons of it. And inside, there's not much. In an effort to try to balance this out, the water that's inside the red blood cells leaves, you know, to water down the solution around the red blood cells. So it tries to leave and go out into the water so that it can bring balance. Again, the problem with this is if it's an extremely hypertonic solution, all of the water leaves the cells, because no matter how much water is leaving, it's still not balancing out the surroundings. So the red blood cells lose all the water. This is something called crenation, and it causes cells to shrivel. Okay, so these are actual colored micrograph images of what happens to cells in these conditions. Notice that, Oakley, my dog, sorry. Notice that cells normally have this nice little bowl shape structure. You can see these little indents. Well, if you look at it, what happens in a hypotonic solution, that indent is essentially gone because these red blood cells have swollen up so much. And then you can see over here in the hypertonic environment that they have these jagged edges, and that's because all the water's left in them. They've essentially shriveled and pancaked out, and they're not living anymore. So a side note, if you've ever heard that you shouldn't drink salt water or seawater, this is why. What happens is, in your brain and in your body, if you start drinking salt water, your blood and the tissues, the fluid in your tissues starts looking like this, where you've got lots of salt, which is a solute, in the liquid and in the tissues around these red blood cells, okay? Lots and lots of salt. Well, we know what happens then. It creates this hypertonic environment where the water from the blood cells tries to rush out to balance it out so it's not so salty, and the blood cells die. This isn't just with blood cells, it happens with other cells and other tissues of your body. So it happens to brain cells, which is what leads to that kind of delirium and insanity that sets in prior to death if somebody's been drinking salt water, because it actually dehydrates you. again, for this reason, what's being dehydrated are the cells themselves as water is coming out of them to try to balance out that surrounding environment. Frankly, this is the same type of thing that you're going to encounter if you are drinking nothing but soda. You're doing the same thing. Sodas are solutions. They're a mix of solutes, different types of sugars and preservative, well, not necessarily preservatives, but lots of different types of sugars and flavorings and colorings and stuff like that. All solutes dissolved in what was a water base. So you're changing the tonicity of your tissues and blood when you drink nothing but soda. Same thing can happen. Soda dehydrates you in the sense that water leaves the blood cells and the cells in your tissues to try to balance out that external environment. Alcohol is especially great at this, which is why the effects of drinking a lot, within a short period of time can be especially brutal, because it dehydrates you so severely, just very severely. Okay, so a couple of practical real life examples of what happens as far as tonicity in your cells. Okay. Move to the next page. So this was an in-class problem set, which, as always, I will ask if you would please complete it, like pause it here and complete this on your own before you tune back in. Okay. So let's look at this. Define diffusion. I've defined it for you, so hopefully you have redefined it here for yourself. Fill in the table below. What they're asking is there a concentration gradient here? Is there a buildup of molecules more on one side than the other? In this case, the answer is yes. I'm going to change my color to this obnoxious pink. Yes, because we've got a lot more on this side than we do on this side. Okay, so the movement for diffusion is going to be this direction as it tries to balance out. Okay, is there a gradient? Yes, it's not as intense as this one, but it is still here. We still have a buildup on this side, which means that we're going to have a flow to the right. And this one, we really don't. We're pretty much even. So we're going to say that we don't have a gradient there and that there isn't any net movement because everything's kind of balanced. Okay, define osmosis. Again, just did that, so hopefully you've redefined it for yourself. What are we going to do in these situations? So intra means inside, so the fluid inside of the cell is a 5% salt, has a 5% salt ratio with water, whereas the extracellular fluid has 10% salt. All right, so basically if I pretended like I've got water and I've got 5 salt over here, actually I don't know why I drew that second one. And outside, I just made 10, okay. I have 10%. What kind of solution is this? Okay, where's water going to want to go? Well, if I'm looking at it, outside of the cell actually has a higher concentration of salt than inside the cell, okay? So this solution is hypertonic because there's more solute outside. If that's the case, where's water going to want to go? Well, it's going to want to go from where there's not a whole lot of solute to where there's a lot. And so water is going to want to go out in this direction. So it wants to, I'm just going to put leave. It's going to leave the cell. This is that crenation or shrinking that happens. Okay. Let's do a different one. All right, we have 87% glucose inside and 74% glucose on the outside of the cell. Well, I'm going to round that, so I'm going to do 9 inside, and then I'm going to do 7 on the outside. Okay, where's water going to want to go? Even though this is not a huge difference, this is a hypotonic solution because there's less solute outside than there is inside. And that means water wants to go in the cell, right? Wants to go in here in an attempt to balance it out so that the outside and the inside have the same amount. With something that has a small margin of difference, then it's likely just going to balance itself out. You know, the extremes of that are the swelling and bursting or the shriveling up and dying as far as hyper and hypotonicity, right? But most of the time, it creates the balance that the body's looking for. Okay, so this where you have 2% inside and 2% outside, 1, 2, 1, 2. This is an isotonic solution. And there's no movement or no net movement. Net movement just means that stuff is still going to come and go, right? Some's going to come in, some's going to go out, but the amount that's inside and the amount that's outside stays the same. What would happen if you drank seawater? Boom, already told you. Talk about crenation, cells shrinking because you create a super hypertonic environment for your blood cells, as well as just the cells in your tissues. Water leaves those cells to try to balance out the salt and the solute in those tissues, causing cell death. A lot of cell death is a bad thing, just in case you weren't sure. You get organ failure. Okay. I'm going to try to go through these without pausing this lecture. Let's see if I can. So let's talk about the plasma membrane a little bit more. This should be a little bit of a review because we've kind of already talked about this membrane a little bit, but let's go over it here. So again, when it comes to the plasma membrane, this is the outer cell membrane for both prokaryotes and eukaryotes, right? And it's called the phospholipid bilayer, again, because of its construction. So it has this consistency of oil. And if you think about that, oil can move, right? It's rigid. So the cell membrane isn't so much a wall, you know, like a brick wall, as it is something fluid that can allow things in and out. And in this case, I don't necessarily mean fluid as in water, I just mean able to move, okay? So it's phospholipid bilayer, the consistency of oil, and it has all these proteins embedded in it. So these are all kind of examples of proteins here. Different types of proteins have different types of functions, but they all, in one way or another, allow different types of molecules to move in and out of the membrane, because if the phospholipid bilayer is going to keep them from moving, they got to get in or out somehow. These proteins help with that. So again, these membranes are selectively permeable, kind of stuck up, right? They only let some things in or out. Okay, that means that only some molecules can freely enter the cell, as in they don't need the help of proteins. They can just come and go as they please without having any assistance. So I'm just going to put only some can move across freely. Okay. Let's talk about some of the things that can get in and out in our little diagram here. So some things that are going to be able to do that. Hang on, let me make sure I've got my diagram situated in my head so I don't tell you this backwards. Okay, all right. So some of the things that can get in are going to be, let's see if I can write it in here, small, like really small molecules that are non-polar. Gases, so things like carbon dioxide, oxygen, things like steroids are hormone building blocks. Those things aren't really going to have trouble being able to move in and out. Some things that are going to have issues are ions and polar molecules. They're going to need some help. So are things like, actually, this one should have been polar. I'm not really sure why when I'm looking at some of these have been written the way they have. Ions, polar molecules, things like water, and something that's large. Large molecules tend to need some help getting across. Okay, so again, what can get in if something is non-polar, for example, carbon dioxide, oxygen, a lot of steroids, and they're very small in size. The things that are going to have a hard time are ions, which again are going to be positively or negatively charged atoms, polar molecules like water, and molecules that are just big, bulky molecules. Okay. So when it comes to these types of things that we just talked about, the ions, polar molecules, and the large molecules, how do they get across? Well, they get across through the use of these proteins I told you about, transport proteins, okay? So when we're using transport proteins, there's a type of movement that's called facilitated. Facilitate, goodness, how do you spell facilitate? Facilitated transport. Okay, so facilitated transport, we're going to have molecules move via diffusion, okay, through protein channels. What that means is, think back to diffusion, things are going along a concentration gradient, right? They're going from where it's like super crowded to where it's not as crowded. In facilitated transport, there's proteins that just let them pass through, kind of like a bridge or a tunnel, actually, or like a tunnel. So the molecules are still going where they want to go, from where it's really crowded to where it's not as crowded, but they're just using the protein as a tunnel to do it. Okay, so that's one type. And then we have an entirely different type of transport that requires energy. So remember I told you that diffusion, osmosis, and facilitated transport, these are all passive types of transport. So I'm just going to write here again. This is passive. The word diffusion should tip you off about it being passive because diffusion is a passive process. But just in case. Okay, passive transport. Active transport, on the other hand, requires the use of energy in the form of ATP, which we're going to be talking about in cellular respiration. So coming up. Active transport is required to move solute from low concentration to high concentrations, the opposite of diffusion. Let's go back to that classroom example I had. Let's say that you are all spread out and I tell you, okay, get up and go to the corner, all of you, and you don't want to go. Or at least some of you were like, that's not going to happen. In active transport, I would, or somebody, would pick you up and physically put you in the corner, right? Because I'm making you go someplace that you don't want to go. And I am using energy to do it. I'm using my energy to pick you up and to move you to where you don't want to go. That's what happens with active transport. These molecules are like, I'm fine here. I'm spread out. I got plenty of room. I don't want to be over there. And so in order to get over there, the cell has to use energy to physically move them. Okay? That's how active transport works. And again, the cell really does its best to not use active transport. The cell's job, one of the many, is to generate ATP, to generate energy. It doesn't want to then use that energy if it doesn't have to. It just made it so that we can take care of our life functions like breathing. So if it doesn't have to use it on transport, it's not going to. So when it comes to transport, then let's talk about bulk transport. In other words, we're going to be moving a lot of stuff at once. So what happens is, in bulk transport, we get something called a vesicle. And a vesicle is this little pouch that forms from the plasma membrane. Okay, so here it is. Here's the plasma membrane. I'm going to say that this is inside the cell. No, this is inside the cell here. So we have something, in this case, it's a quote unquote solid, probably a large molecule that needs in. It's too big to go through transport proteins. That's not going to happen. So what does happen, and this happens chemically, is that a bunch of receptors on the cell membrane bind with this molecule that needs in, recognizes it, and then it creates this little pouch right here. This part of the membrane pinches off and you get this little vesicle. So now the little molecule is surrounded by membrane, part of the plasma membrane. And the plasma membrane, this vesicle, transports it to where it needs to be in the cell before it releases it. Okay, so this is called phagocytosis, or you can think about it as cell eating, mostly because of the size of the molecule. Okay, bring in large foreign matter. So one of the places that you see phagocytosis happen regularly in the body is in white blood cells. Macrophages and other types of white blood cells, you know, when they find a foreign invader, something that doesn't belong there, they engulf it in this process right here that I just showed you. Take it into the cell where it gets dismantled, right? It gets taken apart. And the part of it that causes disease or illness, like if you're looking at a virus or bacteria, that gets saved and passed on, kind of passed around, if you will, to other white blood cells. You're like, hey, look for this thing. It causes disease. I'm going to put eating. Foreign matter, okay? Then what we think about as cell drinking, size and type of molecule, is called penocytosis. Okay, you have the same type of thing happening as far as you have a vesicle break off, but notice that rather than just holding one large molecule, you've got a bunch of small molecules, usually some type of solute that's been engulfed to bring in. So we talked about the fact that water doesn't get across cell membranes very well because it's a polar molecule. So one of the ways that water does get in is through this process of pinocytosis. Okay. All right. And then we have something called receptor-mediated endocytosis. Fancy, I know. Let's see if I can even write it in that room. Receptor-mediated endocytosis. Fun. Okay. So endocytosis, again, is still going to be a process of something entering the cell, but it's receptor mediated. Receptor talking about chemical receptors on the surface of the cell membrane, and usually also whatever needs to get in. And those two receptors recognize each other, okay? And in that relationship, they're allowed to be brought through into the cell. So the solute binds to receptors on the membrane, and they all get brought in at once. See, here you can see that these orange things are going to be the receptors. The particulates, the matter, attaches chemically to those receptors, and then they get brought in. To me, whenever I see this particular image, I always think about escape pods and like a spaceship from the movies. Everybody's in their own space, their escape pod. Everybody's in their own little capsule. That's what I was thinking of. So an example of this is thinking about cholesterol. So lipoproteins and cholesterol, they have to get in and out of the cells, partly because in some cells they're made in there and they have to get out. In others, they have to get in. And an example of this is the LDL and HDL cholesterols that can circulate. We did it. We got through this entire lecture all at once. I usually don't like to do like giant marathon lecture, but to be honest, sometimes it's a little bit easier than trying to break them up into lots of little pieces, especially when these pages are kind of written to one to flow into the other like that. But we didn't. Congratulations. And I will be back with the cell 2, like now.